What is the molecular clock in genetic mutations? Have you ever wondered how scientists can estimate the age of certain genetic changes? The answer lies in a fascinating concept known as the molecular clock. This method helps us measure evolutionary time by looking at how genetic mutations accumulate in DNA sequences. It is based on the idea that certain parts of DNA gather mutations at a steady rate over time. These mutations can be neutral, meaning they do not impact an organism's ability to survive or reproduce. By comparing the number of these neutral mutations between different species or populations, scientists can figure out when they shared a common ancestor. At its core, the molecular clock uses these genetic mutations as markers of time. Mutations are simply changes in the DNA sequence. While some mutations can be harmful or beneficial, neutral mutations accumulate steadily, allowing the molecular clock to act like a timer for genetic changes. The concept of the molecular clock was developed in the 1960s. Researchers noticed that protein and DNA sequences changed at roughly constant rates across various species. By calibrating these clocks with known events from the fossil record or other dating methods, scientists can translate genetic differences into estimates of how long ago species diverged. There are different types of molecular clocks. Some focus on DNA sequences, while others look at protein sequences or specific genes. The rate of mutation can vary based on the region of the genome, the lineage of the species, and environmental influences. Therefore, scientists often calibrate molecular clocks carefully to account for these factors. In the context of family genetic testing and hereditary health, understanding the molecular clock is essential. It helps explain how genetic differences build up over generations. This knowledge is useful for tracing family lineages and understanding ancestral relationships by analyzing mutations passed down through DNA. Practically, molecular clocks assist in estimating when specific hereditary mutations first appeared. This information can help identify the age of genetic variants linked to inherited conditions. Knowing when these mutations arose can inform risk assessments and guide family health planning. So, the molecular clock is a valuable tool that uses the steady accumulation of neutral mutations to estimate evolutionary time. It provides a framework for understanding ancestry, species divergence, and the timing of hereditary mutations, all of which are important for family genetic testing and hereditary health.